Bismillahirrahmanirrahim. My name is Nur Amira and I'll be presenting about extraction of butterfly pea flowers for characterization of antioxidant compounds and beverage making. My supervisor is technologist Dr. Aisyah binti Muhammad Rehan. My co-supervisor is Professor Madia Dr. Amza Sari binti Muhammad Kasim. Chapter 1 Introduction. So for the background study, Clitora, also known as butterfly pea flowers, is widely found in tropical and temperate regions worldwide. It has beautiful deep blue petals, solitary and have light golden borders. In India, traditionally, it is used as a remedy for snake bite and scorpion sting. It can cure menstruation and induce uterine contractions. It also helps with hair growth. But commonly, butterfly pea flowers are used as natural coloring as it contains anthocyanins. Anthocyanins are pigments that give red, violet, and blue colors. The flowers of Clitora contain a variety of phytochemical compounds, including flavonoids, and the predominant coloring components are anthocyanins. Problem statement for this study is because Clitora is often used as natural colorants as replacement for synthetic dyes. This is because the benefits that came from anthocyanin that is present in Clitora. Another reason is because butterfly pea flowers should be made into beverages. This is because another published study on Clitora indicated that Clitora extract display acute in vivo plasma antioxidant effects in herbal beverages. Another problem statement is because a large number of severe diseases caused by free radicals. A free radical is an atom or molecule with an unpaired electron that is visibly unstable. Increasing the antioxidant defense layer in the human body system will eventually aid in lowering free radicals from harming cellular components. The objectives of this study is to perform antioxidant assays of Clitora extract using ferric reducing antioxidant power (FRAP assay) and 2,2-diphenyl-1-bicryl hydroxyl hydrate TPPH assay. Second, to measure reactivity of Clitora extract using UV spectrophotometer. Third, to investigate the effects of Clitora extract as beverages by sample distribution and surveys. Scope of this study is first, Clitora flowers will be extracted to provide a pure extract. Second, Clitora antioxidant compound determination will be performed using ferric reducing antioxidant power assay and DPPH assay. The absorbance of the reaction mixture of the FRAB and DPPH assays will be measured using a UV spectrophotometer to determine the antioxidant activity of Clitora extract. Fourth, the preparation of Kejuruteraan beverage will be tested based on user preferences via survey and sample distribution. Significance of study: Kejuruteraan or butterfly pea flowers are well known for its antioxidant, anti-inflammatory, and other benefits. The identification of effect of antioxidant through beverage samples. The sample of the drinks will be distributed among 30 respondents and respondents will be required to answer a survey. From the survey, the post-effect of consuming butterfly pea flowers, pea beverages can be analyzed. The extraction of Clitora also will be needed for antioxidant assay. We'll move on to Chapter 2, Literature Review. Clitora, also known as butterfly pea flowers, and in Malay we know it as bunga telang. Basically, Clitora has its own natural compounds which can treat disease. It also has different naming in different countries. As for example, in Sudan, they name it as Kordofan pea. In Brazil, they name it as Kunha. And in Philippines, they name it as Bokindang. Butterfly pea flowers comes from family of Fabaceae species. The height is between 90 centimeters to 162 centimeters. It has spiral stems, which can grow up to 3 meters high. The leaves has about 3 to 9 oval-like shape and elliptical. Butterfly pea flowers has small size for diameter of flower, which are at 2 to 4 cm length And apart from that, it has diverse shade of blue with yellow or pure white colorant with big standard petal. It is widely used as natural colorants either for making drinks or into food industries. However, Clitoria also sensitive to temperature and pH changes. But even though they are sensitive to temperature and pH changes, they are excellent in growing either in tropical monsoon country or even at drought area. The benefits of Clitoria tenacia: the effect of antipyretic, analgesic, and anti-inflammatory. It can be a source of food for livestock. It often is consumed by animals because it is mild and pleasant flavor when compared to other forms of legumes. It enhances brain health, which is it increases the levels of acetylcholine in their brain. Acetylcholine levels in the brain have been shown to reduce age-related memory loss and improve memory. Not, uh, not just that, Clitoria also improve one's skin's and hair health. Antioxidant in Clitoria help relax stomach muscles, which aids digestion. It contains anthelmintic properties, which aid in the prevention of worm development in the stomach, regulation of blood sugar levels by controlling the absorption of sugar into the circulation. Relevance from drinking water. Human keeps their water balance by ingesting the same quantity of water as is extracted. The body maintains a balanced level of water by regulating intake and outflow. Negative water balance, also known as dehydration, is described as a decrease in water and salt in various proportions compared to the normal condition, which can be caused by a failure to replenish necessary water losses or a failure of the regulatory system. Dehydration causes hypernatremia, which can be caused by one or more of the following factors: water losses greater than sodium chloride, insufficient water intake, sodium chloride addition, osmotic diuresis, and diuretic therapy when free water intake is insufficient. Drinks, in addition to meeting a fundamental need, are part of human society's culture. Next is activities related to Clitoria tenacia. It is anti-inflammatory, analgesic, and antipyretic activity, anti-diabetic, and antioxidant. It also has phytochemical constituent, which is chemical components, camphorol, quercetin, myristoyl, testosterone, and etc. It functions and phytochemical components in leaves contain alkaloids, flavonoids, glycosides, reducing sugars, steroids. In flower pods, it contains alkaloid, carbohydrates, glycoside, phytosterol, saponin, tannin. In root, DPPH, and in seed, amino acid sequence exact same as insulin. This is DPPH and PrEP mechanism. Well, it has percent inhibition for DPPH and the PrEP value is calculated in micromole TE over mass. Moving on to chapter 3, methodology. For the process flow, the first one is extraction of butterfly pea flowers. Okay, Clitoria tenacia flowers will be collected at a nearby nursery in quantities of 500 grams for the extraction process. For bigger surface areas, the petals will be chopped into little pieces. The petals are then dried for 24 hours at 50 degrees Celsius. The dried flowers of Clitoria tenacia will be weighed after drying. The dried flowers will then be filtered using number 41 Wetman filter paper to obtain only the pure extract. The Clitoria tenacia extract is then gradually dissolved in distilled water at room temperature in a 1 to 25 ratio of sample extract to distilled water. The sample will then be heated at 100 degrees Celsius for 1 hour to release the dye into aqueous solution. After that, the suspension will be filtered. The filtered solvent will be evaporated using a rotary evaporator under vacuum pressure. The dried residue will then be refrigerated until it is used. As for the second one is preparation of making beverages using Clitoria tenacia extract. Each formula will be mixed with distilled water to make a 250 milliliter of butterfly pea extract drink. Surveys are then used to analyze the samples among 30 respondents. And then the third one is distribution of beverages samples and surveys. Okay. Beverages samples and surveys are distributed simultaneously, meaning that once the beverages samples are prepared, then the respondents are required to fill in the survey simultaneously with the drinks provided. As for the third one is for antioxidant assay, it is ferric reducing antioxidant power, also known as FRAP. Okay, FRAP assay relies on the reduction of the ferrum 3 TPTZ complex to the ferrous form at low pH. The reduction is tracked by measuring the change in absorption at 595 nanometer. 200 microliter of Kejuruteraan extract will be combined with 1.3 milliliter of prep reagent. After 30 minutes of incubation at 37 degrees Celsius, the absorbent will be measured at 595 nanometer with a spectrophotometer. The FRAP reagent will be made daily and comprise of 0.3 molar acetate buffer, 10 millimolar TPTZ in 40 millimolar hydrochloric acid, and 20 millimolar ferrum chloride in a 10 to 1 to 1 ratio. As for the last one is DPPH, radical scavenging assay. This antioxidant activity of butterfly pea extract will be measured using spectrophotometer after 30 minutes of incubation at 25 degrees Celsius, protected from light. 100 milliliter of methanol containing the test compound in varying concentrations ranging from 0.25 to 5 milligram per milliliter will be added to 100 milliliter of 100 millimolar methanol solution of DPPH, and the absorbance at 517 nanometer will be determined. So for chapter 4 is expected result. So expected result for this study is pure extract of Kejuruteraan is obtained that can be used for future antioxidant beverages. Extraction method will be crucial procedure to ensure that the subsequent process can be achieved. Providing enough amount of 80 gram of butterfly pea extract, enough for each sample. It can be distributed among 30 respondents and surveys will be given simultaneously. 85% of the respondents will love the taste and color of Kejuruteraan extract beverage. And then, in this study, it can perform the antioxidant assays using DPPH and FRAP. It measures the content of antioxidant compound in the Kejuruteraan extract and the absorbance of the reaction mixture of FRAP and DPPH assay will be determined using UV-based spectrophotometer. This is the Gantt chart from week 1 to week 16 and the list of activity that had been done. Alhamdulillah, I have designed all the slide which is started from introduction to the expected result for my study, which is extraction of butterfly pea flowers for characterization of antioxidant compounds and beverage making. I think that is all from me. Thank you.